Welcome to the Dani Research Group. Our current research is on smart microwave and millimeter wave sensors for water detection, the electrical properties of natural elements such as plants, soil, etc., and the effect of microwave radiation on plants and animal tissue cell dynamics. My project is on the calibration of a vector network analyzer with the open short load method. A vector network analyzer, or VNA, is an instrument that measures the response of any circuit using electromagnetic waves transmitted and reflected from the circuit. We use the VNA to measure dielectric properties of a material, which define the electrical field dissipation within the material itself. These properties are useful in determining moisture content in plants and soil with in-contact measurements. We have done precision calibration of the VNA using dielectric probe kits and various loads, such as deionized water, alcohol, and saline water, to determine the measurement accuracy. The pictures on the right are just kind of an example of how our tests were set up. And with our test, we found that deionized water gives the best results in terms of broadband load match from 500 megahertz to 20 gigahertz. This means that deionized water produces the least amount of error compared to alcohol and saline water.
My project is on the planar antennas for passive radiometric soil moisture measurements. So as a group, we have been working toward developing passive and active sensors to detect moisture content in plants and soil using radio frequencies, along with studying the effects of electromagnetic radiation in the frequency range of 100 megahertz to 20 gigahertz in botanical and zoological tissues. Our goal is to design a planar, compact microstrip PCB-based antennas to detect blackbody radiation from wet or dry soil for the goal to reduce water waste in agriculture. We are able to convert the data from the antenna into a soil moisture index through radiometric algorithms. And we are using a 3D electrical magnetic simulation tool called CAD-FECO to design and develop specifications for our patch antenna that we plan on building in the future.
My research project focuses on testing the effects of RF microwave exposure on plant germination and growth. This is done by understanding the biological effect in plant tissues when exposed to RF microwave radiation in the frequency range of 500 megahertz to 20 gigahertz. We do this by using seed samples with in-contact exposure to a VNA and a coaxial probe for different timelines ranging from 30 minutes to about 2 hours of exposure. We are exposing Coriandrum sativum, or coriander seeds, to differing frequencies of MMV radiation and testing the effects of radiation on germination speed, shoot length, plant output, and other physical distinctions. DNA from seeds focusing on the CO1 mitochondrial plant DNA marker, as well as protein content, will be analyzed. And these results can determine if microwave radiation techniques can lead to a greater product yield overall.
As my other group members have been working with plants, my project is revolving around working with cancer cells. More specifically, our goal is to find the difference between cancer cells and normal cells, utilizing radio waves to find their different dielectric properties. Dielectric is having the property of transmitting electrical force. In the medical field, the term is often used to describe the permittivity of tissue for treatment that uses electromagnetic waves. Every type of tissue has a different permeability, and tumor or cancer tissue has a unique measurement as well. These dielectric properties vary between different types of tissue depending on the water content and makeup of the tissue. For example, fat has a much lower permittivity than brain or cardiac tissue. Fat does not hold much water compared to tissue from organs, so the lack of water leads to lower permittivity and a higher reflection coefficient. All these properties are then used to determine the type of tissue and any issues the specific area of tissue might have. We plan to use a vector network analyzer and coaxial probe to measure and collect the dielectric properties to differentiate cancer and healthy tissues.
My project is to study the effect of radiofrequency or RF radiation on animal tissues. This is to understand the biological effect in animal tissues when exposed to RF radiation in the frequency range of 500 megahertz to 20 gigahertz. Tissue culture samples will be used with in-contact exposure using VNA and a coaxial probe for different timelines ranging from 30 minutes to about 20 hours. Biological changes in the sample will be determined by performing DNA tests using PCR machines, protein synthesis, and visual inspections under the microscope. Thank you.